This is Unit 2, Part 3. There are four important aspects or concepts that are important to understand about the cell. One is it's the basic structural and functional unit of all living organisms. The second is that the activity of a living organism will depend on the individual and collective activity of its cells. When we study multicellular organisms like humans, it's the collective activity overall that's important, and how you get specialization and interaction. The third thing is that biochemical activities of cells are dictated by subcellular structure. The structure underlines function, and that includes biochemical activity. And the fourth is that continuity of life has a cellular basis. So in order for an organism to reproduce itself, it uses cells in the reproductive process and allows for continuity of life. There are many cell shapes. You name it. You don't have to memorize these. These are just sort of examples. We will talk about cell shape in more detail when we do histology, and you'll learn quite a bit of histology, particularly in the lab. But for now, there are many possible shapes for cells in the body, which, by the way, are characteristic of different cell types. Each cell type has a characteristic shape. Three major parts of all cells are the plasma membrane or cell membrane, which surrounds the entire cell, the cytoplasm, which is the soluble liquid portion containing quite a few organelles inside it, and the nucleus, which is also surrounded by a membrane and contains the genetic information coding for the organism that the cell is in. And here's a diagram of the cell. It's sort of a generalized diagram of a cell. There's many structures here, and I'll go through them, probably almost all of them, or all of them. And not all cells necessarily look this way, but cells contain these structures. And so it's a sort of a generic kind of a cell diagram, and is useful for us to look at in order to understand the subcellular structure and function. We're going to go through this one at a time, but suffice it to say that the cell membrane or plasma membrane is the exterior membrane that completely surrounds the cell. The cytoplasm is between that and it's located between the plasma membrane and the nucleus. So everything between the outer plasma membrane and the nuclear membrane is the cytoplasm, and the cytoplasm contains many organelles, membrane-bound organelles. Okay, as I said, the plasma membrane is the same as the cell membrane, separating intracellular from extracellular fluid. It plays an important dynamic role in cellular activity, and that means that it is involved in movement of molecules across the membrane, both in and out. And it's selective what it does allow in or what it does allow out. For most molecules, some it excludes, others can pass through easily, but many are regulated in their passage through. There is an area outside of the cell membrane which is called the glycocalyx. It's on the surface. It's an area that has typically molecules in it that are part protein and part carbohydrate, and therefore are called glycoproteins, because they're part polysaccharide or carbohydrate, and they are part protein. That outer area has molecules, glycoprotein molecules on it that act as highly specific biological markers or molecules, biomolecules, which help cells interact with each other or recognize one another as self. It's very important in a multicellular organism that the cells are able to recognize self from non-self. You know, you have to understand that in a multicellular organism you have specialization of different cells, and they can communicate through each other with each other, often by secreting a molecule. If another cell has the receptor for that molecule, you know, on its surface, then it can respond to that signal. Now, this model that I'm going to describe for the cell membrane is called the fluid mosaic model. The cell membrane is a phospholipid bilayer. It's 2 layers of phospholipids, and embedded in them, in that layer, and associated with the layer are different kinds of proteins, often glycoproteins, but proteins, sometimes glycolipids. Glycolipid, of course, is part carbohydrate, part lipid. So the vast majority of the phospholipid bilayer is, in fact, phospholipid, but there is also quite a bit of cholesterol, and that adds fluidity to the membrane, and there are some glycolipids, as I said. And as I told you earlier, glycolipids are lipid that have a portion of them is carbohydrate. The phospholipids are partly hydrophobic and partly hydrophilic. The phosphate, and we talked about this when we described phospholipids as a type of lipid. The phosphate part of the phospholipid molecule is hydrophilic, and the fatty acid chains in the phospholipid molecule are hydrophobic. So here we see a model, and it's extremely similar to the original paper that described this by Nicholson, which was published a long time ago. Not that long ago, but a long enough time ago. And the original paper is the diagram in that is very similar to this diagram here. Here you see we're looking at the outside of the cell, the extracellular part, and here we're looking at the cytoplasm on this side. Remember that living things use water as a solvent, so the extracellular fluid will be water-based, and the intracellular cytoplasm will be a water-based solution. You have phospholipids facing outwards, and it's the phosphate part of the phospholipid molecule that faces outward, because the phosphate part is polar and can interact and be stable in its interaction with the water in the extracellular fluid. Inside you have the 2 fatty acid chains. I'm going to highlight them here. So with this let's do it in red. So here, this molecule, there's 2 fatty acid chains associated with the phospholipid head. And here again, 2 fatty acid chains associated with the phospholipid head, right? So what you have are the fatty acid chains, which are hydrophobic, facing inward, and the phosphate part, the polar hydrophilic part or head of the molecule facing outward. And then you have the second layer, which faces inwards. Similarly, the phosphates in this case are facing towards the cytoplasm, and the internal part, the fatty acid chains, to the lipid part of the molecule are facing inwards, and so what you have between these two this phospholipid bilayer is a hydrophobic region. Now, embedded in the hydrophobic region are proteins that I'm outlining one there. Maybe I'll do it in a different color and I'll highlight it in this way. What you have are these kind of different types of proteins embedded into the phospholipid bilayer cell membrane. Sometimes they go right across, sometimes they're just partly embedded and don't go across. Sometimes they're only associated with the membrane on the inner side. These are called the ones that are just, and sometimes they can be associated on the outside. These are called peripheral proteins, okay? Because they're not embedded in the membrane, they are associated with it. Just a second. There are also some of these embedded proteins pass right through, and there are many different functions for different specific proteins that are found in the membrane. Some even act as channels for passage of specific molecules, and see, there's a channel going through. So if this is open, then the molecule that it allows through will go through inside, right? Some of the proteins have a carbohydrate portion to the molecule. There's this carbohydrate. This is a diagram of several monosaccharides strung to get linked together by covalent bonding to show that it's a polysaccharide attached to this protein molecule. And if you have a carbohydrate attached to a protein, then it's called a glycoprotein. In this case here, beside it to the right, you have a glycolipid because you have a carbohydrate molecule here attached to lipid here, these fatty acid chains, they're lipid. Okay. You should notice also that throughout the membrane, scattered throughout the membrane, is quite a bit of cholesterol. It's an important part, an important component of phospholipid bilayers. And as I told you before, it adds a certain degree of fluidity so you can get movement of the proteins in the membrane. They can move around. I believe I've covered most things on this slide that I wanted to. Again, I just want to point out to you that this area here in between is made up of non-polar, hydrophobic, uncharged molecules. So it's a hydrophobic area. And the phosphate heads here and here are hydrophilic, and they can therefore, when it faces outwards, it adds for stability. There's tremendous stability in this, even though the protein molecules can flow through it, and it does have some fluidity to it. It is a stable structure. Okay. Now, different cells might have different lipids in the membrane, different proteins. The inner face of the phospholipid bilayer does not typically have the exact same phospholipids as the outer face. There are differences. Glycolipids are found only in the outer membrane, not on the inner membrane. And a surprisingly large amount, as I mentioned, a significant amount of the membrane lipid is in fact cholesterol. As I said, integral proteins are embedded in the membrane, whereas peripheral proteins are associated with it. And most peripheral proteins are associated on the inner side, but there was an example in that diagram of one that was associated with the membrane on the outer side. And these peripheral proteins, typically, they're associated with a protein that is embedded. Again, there is this outer glycocalyx area of carbohydrate-rich outer area at the surface, composed of both glycoproteins and glycolipids, and the diagram showed that in that it showed these carbohydrates extending away outwards from the some of the proteins embedded in the membrane, and also extending away from the free fatty acids that were in the phospholipid bilayer. The membrane at certain points has extensions to it, and these are called microvilli. And microvilli are not found on every type of cell. Microvilli are particularly important in cells that need to have a large surface area because they're involved in absorption, and so the bigger the surface area, this membrane surface area, the better they can absorb. And a typical example of cells that would have microvilli at their surface would be cells that line the surface, the exterior surface in the intestinal tract, where they are involved in absorption. So these are like finger-like extensions of the plasma membrane at the surface. Now, joints or junctions between cells are held together by different types of structure. One is what's called a tight junction, and it completely encircles the cell. So if we drew a cell, and remember the cells are not in 2 dimensions, but in 3 dimensions. So if I'm gonna just change this, hold on. So if we draw a cell in 3 dimensions, what you have here, if I'm drawing it correctly, no, hold on. Please be patient. Let's look at a cell drawn in 3D. So if you have a cell there and then you have another cell beside it, right? Maybe my ability to draw is limited. Hold on. Maybe we'll just go to the diagram on the next page, on the next slide, which would be better. Let's forget this. Let me just describe this and then describe it on the diagram. It's a lot easier. Those diagrams are a lot better than anything I could come up with. Okay, so tight junctions are found all around the sides of the cells, between one cell and the next cell, and they are very impermeable. Another type, and we're going to go through each one, describing each one in the next few slides. Another type of junction between cells is desmosome, which acts to anchor, again along the sides. And then another type of structural junction between cells is called a gap junction. And that allows for communication between the cells because it allows for chemical compounds to pass easily between the cells. So here you see cells drawn, as I said, much better than I did in 3 dimensions. So this is the side wall here of the cell. And what you see when you look at the structures called tight junctions is that they are around all the way around. If you look at, they don't show them on the back, but they're there. They don't show the front, obviously. They've taken that away so you can view into the cell. But these structures are all around the cell and all around the next cell, and they interact with each other. They're proteins that interlock with each other. So these are tight junction proteins, proteins that form a tight junction and are bound very, very tightly to each other so that things can't get in. If this is a surface epithelial cell, things cannot get in between, okay, and get into the tissue underlying. If this is the exterior of the body, you have to be very clear that the exterior of your body is anything that is continuous with the outside world. So outside of your skin, which is an epithelium, that's outside, and that epithelial surface curls around and goes down your mouth and down your esophagus and your entire gastrointestinal tract. So the space inside your gastrointestinal tract is external to your body tissues. It's outside of your body. It's outside of your body tissue. And what's in there, you don't want to get into your body tissue. You don't want the contents of your intestines to get in between the cells and then into your body tissues. It's full of bacteria. So there are tight junctions which form a barrier. As you can see, there are lots of microvilli. I told you before here, microvilli. I told you before that you find microvilli in the lining of the cells that line the intestine because the intestines are involved in absorption of nutrients. And you find tight junctions. And here you see the other type of junctional structure, membrane junctions, which are desmosomes that help anchor across, and they have these extensions going into the cell. And here is a gap junction, not the greatest diagram. Perhaps we'll see a better diagram. Anyhow, here, this one is tight junctions. The next one is the desmosomes, and you can see how well they anchor across. And these are all protein structures helping to anchor two cells together and go all the way in. And then you see, and this is a much better diagram, here you see gap junctions. Now there are gap junctions between many different types of cells that are adjacent to each other, but an extremely important area where you have gap junctions is in the heart, in heart muscle, because heart muscle cells have to communicate with each other so that they all contract together at the same time. And that is possible because these protein structures, gap junction structures on one cell, I'll highlight it here, that's one, and there's the other. They align very nicely with each other and form a channel so that molecules can in fact go straight across either way. Okay, so we have tight junctions, desmosomes, and gap junctions. Now, cells have to have some sort of transport mechanisms across the cell membrane. As I told you before, not everything can get across. It is a selectively permeable membrane. The plasma membrane is selectively permeable. There are a number of different mechanisms that allow certain molecules to get across. A few molecules can move across and are not stopped by the presence of the membrane. Others have to be interact with specific receptors before they could move in, or molecules that will open up and form channels to allow them in. Some transport mechanisms require energy, and those are called active transport mechanisms, and some are passive, don't require any energy input. They're passive. Let's talk about the passive ones first. Simple diffusion, facilitated diffusion, and osmosis are all passive transport mechanisms that do not require energy. The movement is strictly due to differences in concentration, and that's on one side of the membrane versus another. That's called a concentration gradient. For some molecules, they will be able to move simply by diffusing across, simple diffusion. And examples of that would be oxygen or carbon dioxide, or you might think water, but movement of water is called osmosis. Movement of water across a semi-permeable membrane is called osmosis. And actually it does require pores that some cells express. Let's not get into that. So simple diffusion does occur. All lipids can go across because they're not stopped by the phospholipid bilayer, even though it's got a hydrophobic middle, a non-polar hydrophobic middle region of fatty acids. Small fat molecules lipid molecules can move across it because they are also lipid soluble and will move across. Gas molecules in solution like oxygen and carbon dioxide will move across. And the driving force again is the concentration, always from higher to lower. Okay. Facilitated diffusion also doesn't require any energy. Will go from higher to lower concentration areas. However, it does involve specific carrier proteins. Okay, so there is a specific carrier protein involved for each of those. And then the diffusion of water, okay, diffusion of water is given its own particular name. It's called osmosis. So it's not, when we talk diffusion, we're talking about other types of molecules. When we talk diffusion of water, we're talking about osmosis. And again, actually, when you think about it carefully, it is from higher concentration of water to lower concentration of water. And it's a movement of water across a selectively permeable membrane, which, of course, a phospholipid bilayer, the cell membrane, is. Now, there is one other type of passive movement called filtration, and filtration is important, particularly in the kidneys. The kidneys carry out filtration of the blood plasma. Blood goes into the kidney, the plasma is filtered, and a fluid called the filtrate goes into a tubular system and gets processed into urine and out. The driving force is the hydrostatic pressure of the blood. Okay, so I talked about each of those, but we'll go through each of those here on this slide. All right, so we'll go through each of the mechanisms in turn that I just introduced. In terms of passive transport, diffusion, simple diffusion, is the movement of non-polar lipid-soluble substances across the bilayer. Nothing prevents them from moving across. They just move straight across, all driven by concentration gradient. They go from higher to lower areas of higher concentration to areas of lower concentration. So they can diffuse directly. Some do diffuse through channel proteins, but most would diffuse directly through the lipid bilayer. This is an example of simple diffusion. There's no membrane involved here, but what you have is a solution of water on the left and a colored substance that's in solid form, and it's put in and it dissolves into the water, and with time it spreads throughout, going from high concentrations to lower areas of concentration, just spreading throughout simply because it's going into solution and being bombarded by molecules and moving. Now, facilitated diffusion involves movement, passive movement across membrane of molecules, and it doesn't require energy. And again, it's from high level to lower areas of higher concentration to lower concentration. But there is a specific carrier protein involved. So they're either binding and moving across because of binding to carrier proteins, or moving across through a protein that's acting as a channel, but it binds and moves through. Now, these carrier molecules are typically integral proteins. They're transmembrane. They go right across. Then they have specificity. And of course, it's going to be specific for polar molecules. Polar molecules can't move across without some sort of carrier. They can't get across that non-polar inner hydrophobic region. Typically, the kind of molecules that move across through facilitated transport involving specific carrier molecules would include carbohydrates and amino acids. And here you see a model showing a phospholipid bilayer across here with the phosphate groups and the inner non-polar region. And these are lipid-soluble molecules in solution here that can move straight across because they are lipid-soluble. They are typically lipid themselves, or there might be some molecule that is quite lipid-soluble. This is simple diffusion. Another is channel-mediated diffusion, and this is channel-mediated facilitated diffusion, where you have a channel that allows the molecules to go across. In this way, polar molecules can move through the phospholipid bilayer because of this channel protein. So the channel protein allows for a facilitated diffusion. And in this case, these are lipid-insoluble molecules. They cannot move through this phospholipid bilayer in this area here. They need the channel protein. And then there's carrier-mediated. So there's channel-mediated and carrier-mediated facilitated diffusion. And here you have a carrier molecule interacting with the lipid-insoluble molecule, helping transport it across, but this does not involve any energy, but there is a specific binding. These are specific proteins that bind, so the molecule binds in here and is allowed across, as shown here. Here it is crossing into the cell. And then there's simple diffusion due to, with molecules like oxygen or carbon dioxide out, which again, it's just simply concentration gradient is not stopped by the phospholipid bilayer. A special kind of passive transport involves the diffusion of water across the semi-permeable membrane of the cell. The cell membrane is semi-permeable. It's not permeable to everything. It is semi-permeable. And diffusion of water occurs quite easily. The membrane does not stop it, but it's again it's a concentration driven, in that if the concentration of the solvent, which in this case is water, is higher on the outside than the inside, then the water will move across. And this does happen, that you can have the concentration of water higher outside than inside. If the inside has more things dissolved in it, then water will tend to move across. The degree or concentration of molecules dissolved into the solvent, the water, is called the osmolarity of the solution. And the ability of a solution to cause a cell to either gain water or lose water, in other words, for osmosis to occur in or out, is called tonicity. So if you're talking about an isotonic solution, that's a solution that has the same osmolarity inside as outside. If it's hypertonic inside, if the surrounding solution around the cell is hypertonic, then water will move from the inside out to try and reduce it. Or if the solution is hypotonic, lower outside than inside, then water will move from the outside in. And here's an example of movement of water, diffusion of water across a semi-permeable membrane, and therefore, in other words, osmosis. And here you have the semi-permeable membrane. They're showing that it has pores in it. They're exaggerated in size, obviously. On one side, you have a solution that is a higher concentration. It happens to have more sugar in it than carbohydrate in it than on the other side of the membrane. And so movement, and it does not allow these solute molecules across. It's semi-permeable, so it doesn't allow the solute molecules across. Excuse me. In this particular example, it does allow solute molecules across. And not only will the solute move across from areas of higher concentration to lower concentration by diffusion, simple diffusion, but also the water will move across in the opposite direction by osmosis, because water will be drawn across because of the hypertonic nature of the solution on this side versus that side. So water moves across this way, and the solute will move across the other way until the two are balanced and you have equal concentration on both sides and they both have the same osmolarity. And here the volume is unchanged because everything can move across. It is a membrane, not semi-permeable as I said in the beginning, but permeable to both. I made a mistake. I'm sorry. I said initially semi-permeable. It is permeable to both, and therefore they each move across in opposite directions. And you get no difference in volume on either side. So they're both moving across that permeable membrane. The next slide will show what happens when you have a semi-permeable membrane. In this case, it's semi-permeable. Solute molecules cannot move across, so it's impermeable to the solute, but of course permeable here to water, and therefore only water moves across until the tonicity is balanced on both sides, or tries to at least move across until tonicity is balanced, concentration balanced, and therefore water will increase on this side by osmosis, and you get a greater volume here of water on this side than on this side. That's movement across a semi-permeable membrane. Permeable to the solute, but not to the solvent. It's permeable to the solvent, water, but not permeable to the solute. The number of these solute molecules, notice, stays the same. In other words, the concentration here is higher because the volume's smaller and water is moved away from there, but the actual number of molecules is the same as before, or should be. If the number, they're showing the number of molecules being the same and water moving across, so you start getting a higher concentration here than it was before, and the volume on that side is higher than it was before. Okay, another, and I believe the last type of passive transport that we'll talk about is filtration. In the case of filtration, water and solutes will move through a semi-permeable membrane because of hydrostatic pressure. Okay, there's a certain pressure on one side higher than the pressure on the other, and therefore there's a pressure gradient, and that helps push the fluid across from higher pressure to lower pressure area. So the solute containing fluid will move across. Some solutes can move across, the water can move across. Some solutes are not able to move across. In the case of example of a kidney. In the end, we should always look at solutions in terms of their tonicity. There's isotonic, as I said earlier, hypotonic, and hypertonic. Isotonic, the solution has the same solute concentration inside the cell as outside the cell. So the cytosol, the fluid of the cytoplasm, is the same as outside the cell. So it's an isotonic solution outside of a cell. A hypertonic solution outside of the cell, you have a greater solute concentration than that inside the cytosol. Hypotonic, the solution is less than that of the cytosol. In this case, if there will be movement, but it'll be equal in both directions. Okay, the osmosis will be equal in both directions. In this case, what you'll have is water moving out of the cell, not in. And here you'll have, for a cell in a hypotonic solution, you'll have water moving into the cell. So this is into a cell, water will move in, and here it will move out. Okay, and the driving force is the concentration gradient. And this is an example, 3 images of red blood cells for showing examples of what goes on. One is a red blood cell, which is in a isotonic solution, this one. And so it retains its normal shape. That's an isotonic. The cell is in an isotonic solution. In this case, it's in a hypotonic solution, and therefore water will move in, and the cell expands and eventually will rupture. It'll lyse. You'll get lysis of the cell, bursting of the cell. And in this case, in the middle, this is something that happens only with red blood cells. They will pop into this particular shape, okay? You get this sphere shape with spikes on it. That's because a red cell normally has this biconcave shape. A normal red blood cell is like a disc that's concave on both sides, both of the flat sides of the disc. If it's in a hypertonic solution, what happens is that water will move out of the cell through the membrane and out, and the cell will pop into this sphere shape with spikes on it. Here you see all these spikes. Here's a spike pointing right up towards the camera. And this process, or this shape, is called crenation. And you should remember this term, crenation. Okay, so crenation is the change of shape of a red blood cell when it's in a hypertonic solution. So we talked about simple diffusion. We talked about passive types of membrane transport. So we talked about simple diffusion, facilitated diffusion, osmosis, and filtration. The energy source for simple diffusion, facilitated diffusion, and osmosis all involves concentration gradients. The movement of an example of simple diffusion would be movement of oxygen through a membrane. Facilitated diffusion example would be movement of glucose or amino acids into a cell aided by a transporter channel, a specific type of transporter molecule. Osmosis is the movement of water in and out of cells through that semi-permeable membrane. And in the case of filtration, the driving force is the actual pressure gradient, the hydrostatic pressure. And that forms what's called a filtrate in the kidneys. And that's the end of Unit 2, Part 3.